Hello, this is Gina Piscatelli with a lecture on the skeletal system. It's the second lecture, and we're going to be looking at how bone is formed. There's two processes, intramembranous ossification and endochondral ossification, that we will examine. Now you may recall that ossification, the formation of bone, occurs at different points in our lives. The first time, of course, is when we're an embryo and a fetus, and that's when bone development occurs. But after we're born and we grow in height, our bones elongate. So that's the second time point. But throughout life as an adult, post age 26 approximately, our bones can't elongate, but they can widen somewhat. And so this ability to widen our bones continues throughout life. And it's actually a part of a process that also occurs throughout life, and that is bone remodeling or repair. So this lecture is only going to deal with bone development, not elongation, widening, or remodeling. So during development, embryonic development, before week 8, the embryonic skeleton is laid down or formed, and it's composed of fibrous connective tissue membranes and hyaline cartilage, so very soft. Over time, that is replaced with bone tissue. There's two different methods that I mentioned, intramembranous ossification and endochondral ossification. Intramembranous will occur to form the flat bones of the skull and the clavicle. And in this case, that fibrous connective tissue membrane is going to turn into bone. The second method, endochondral ossification, you start with a hyaline cartilage model, and then bone replaces that. So the cartilage dies and bone forms in the same shape as the model. This will occur to form all the bones below the base of the skull except the clavicles. So long bones, short bones, and a few flat bones and irregular bones. Not the flat bones of the skull and not the clavicles. So we're going to start with that one, intramembranous ossification. We begin with kind of a generic embryonic mesenchymal tissue. It's really a fibrous connective tissue membrane made up of cells called mesenchymal cells. And there's a series, I should say, of different steps that turn this fibrous connective tissue into bone. And these are the steps. First, an ossification center is going to form in the center of this mesenchyme membrane, somewhere about here. And the first thing that ossification center does is lay down bone matrix, but it's not hard bone matrix. It's called osteoid. It's soft. Then that osteoid becomes hard when minerals, and calcium in particular, are deposited onto it, and it hardens. Then spongy bone will form, and then a layer outside the forming bone, the periosteum, will form from mesenchyme, and then compact bone forms. So here we have a mesenchymal membrane, connective tissue. We'll say it's up in the skull region. And some of the cells that were mesenchymal cells, they turn into osteoblasts. They differentiate. And so these cells now are blastic, meaning they can cause growth. And where they all aggregate together, that's called an ossification center. So in step 2, these osteoblasts release or secrete osteoid. It's mainly collagen. Okay, it's thick, maybe viscous, but it's not hard. And it's shown here as kind of this yellow material. Now, surrounding this mesenchymal membrane as well as the ossification center, there are some embryonic blood vessels forming, okay, that I tried to draw. There's red blood cells inside there. So the bone doesn't form by itself. The skeletal system doesn't form in isolation. There's many other organ systems developing at the same time. And these blood vessels explain where the minerals can come from. So step 3 is mineralization of the matrix. And that is that calcium and phosphorus or phosphate minerals are deposited into the osteoid tissue or the osteoid material. And that hardens the matrix. So the calcium and the phosphorus comes from the blood, and some comes from the osteoblasts, and it hardens the matrix. Now, on the surface of this ossification center, there are osteoblasts that didn't get trapped by the hard matrix. In the center, there are osteoblasts that did get trapped. Okay, now those are called osteocytes. But the osteoblasts that can still lay down soft matrix are on the periphery, and they continue to lay down more osteoid around the blood vessels that are forming. And so we get patterned, kind of an inner lattice work pattern of trabeculae of bone. So this is a trabeculae, this is a trabecula, this is a trabecula. Next, the mesenchyme that's superficial to the osteoblasts turns into periosteum, okay, which is just a dense connective tissue, but it helps protect the bone. And what will still happen is that these osteoblasts on the surface can still deposit osteoid. So you can get a layer of compact bone forming in this region, and the blood vessels bring in the minerals. Now, since there was blood vessels winding all the way through these trabecula, there are many types of stem cells floating in the blood, and those stem cells will populate the trabecula and red bone marrow will appear. That will include hematopoietic stem cells. And hematopoietic stem cells will become red blood cells or white blood cells and enter the bloodstream. So that's called intramembranous ossification, and it starts with a membrane of connective tissue. It's really not exactly dense connective tissue, but it's a membrane of embryonic mesenchyme. And what happens is first a primary ossification center occurs or develops, and the osteoblasts lay down soft matrix. That soft matrix, the osteoid, is mineralized and it hardens, and some cells become trapped inside that hard matrix. But there are still cells, osteoblasts, that make more osteoid tissue, increasing the width of the spongy bone. Mineralization follows. Then the periosteum forms on the outside when it's wide enough, and then compact bone typically gets a little bit thicker here at the edges. Okay, so that was intramembranous ossification. Now we're going to look at endochondral ossification. This occurs in all bones, as I mentioned, inferior to the skull except the clavicles. Okay, now bone is going to form from models of bones, precursors to bones. They're small, but they're the right shapes. They're called hyaline cartilage models. They develop from the same embryonic mesenchyme that we looked at before. Development is quite complex. Everything is very amorphous to begin, and there's many different stages of development or differentiation. So this hyaline cartilage model of each bone is surrounded by a connective tissue layer called the perichondrium. Ultimately, what will happen is that the hyaline cartilage model will grow. It will increase in length on both ends. The cartilage will be destroyed from the interior outward, and bone will come in and replace it. So that ultimately, when we get to at least adolescence, a bone is going to consist of just one region of cartilage still remaining, and that's called the epiphyseal plate. Usually there's also articular cartilage on the ends. But we're going to start here with the hyaline cartilage model, surrounded by perichondrium. And the perichondrium contains stem cells, mesenchymal stem cells, that eventually turn into osteoblasts, and they're shown in blue here. So these osteoblasts are underneath the perichondrium or embedded within it. That's step one. So step two, what happens is these osteoblasts start laying down bone tissue, osteoid, right? And that hardens. When that occurs, a bony collar forms. So usually the osteoblasts are just active in the shaft region of the bone. So we get a bony collar. In this region now, the perichondrium is called periosteum. It's changed. Okay, so there's a periosteum lining on the outside as well. Now, as you can imagine, if there's hard bones surrounding here, and there's no blood vessels permeating yet, it's going to be hard for materials to diffuse into the center of the cell. The model. But as this bony collar is developing, becoming ossified, growing in length, there are chondrocytes that are in the shaft, and they continue to undergo mitosis. So you're getting hypertrophy of tissue, and so you're getting growth of the cartilage model. So the cartilage model starts growing at the primary ossification center. So it's going to grow that way, and it's going to grow that way. As the chondrocytes lay down minerals in the matrix, it gets harder and harder and harder. So where this is occurring is called the primary ossification center. Kind of weird. We've got chondrocytes laying down osteoid. So the calcified matrix and the bony collar has cut off any diffusion to nutrients, and it's grown really big too. So right in the center where the ossification center started, the chondrocytes die and the matrix deteriorates. And that forms a cavity, which we know will eventually stay in the adult bone as the medullary cavity. But there's still growth happening at the very ends. Eventually these ends will die, just like where the starting point was, but not yet. Then a rescuer comes in, and it's called the periosteal bud. The periosteal bud is going to try to save this bone from death, right? So what happens is the periosteal bud, which is a bunch of things that one of the things is a blood vessel, shown here, that can penetrate the bony collar. It has the right enzymes to break down a little tunnel and get the blood vessel into that cavity. Okay, that is going to bring nutrients, oxygen, but maybe more importantly, some osteoprogenitor cells that will lay down spongy bone in the cavity. So spongy bone will be laid down. And the blood is also bringing calcium and phosphate, so the osteoid that's laid down will harden. Now there probably is some debris of the cartilage matrix left over, and the osteoclasts can break that down. So osteoclasts come in with the blood, as do osteoprogenitor cells. Osteoprogenitor cells are going to become osteoblasts to lay down bone, but the osteoclasts are going to break down the old cartilage. Clean up, basically. In the last step, what happens is two secondary ossification sites form in response to receiving some blood, and this occurs in the epiphyses of the bone. And first you'll see cartilage growth, then cartilage death, and then with the blood comes in the osteoprogenitor cells, so that we lay down spongy bone and bring in the minerals to make it nice and hard. So in the end, with these two ossification centers, the secondary and the primary, eventually meeting, this one's going to kind of grow this way and this way. This one's forming this way. You're going to end up with just a little region that persists as cartilage and cartilage matrix. So the results of endochondral ossification are that the epiphyseal plates right here will remain until mid to late 20s, right? And then eventually that will become bone. The hyaline cartilage is laid on later by a different process. I mean, the articular cartilage at the ends of the bones, that's put on by a different process later, and of course that stays for life, except for the degeneration due to age. So overall what we saw was in endochondral ossification, we have a cartilage model, a hyaline cartilage model, and a bony collar forms by osteoblasts. That cuts off nutrients in the center of this cartilage model, which is growing at the same time. So it's getting bigger and bigger, okay, both directions. But that's going to cause degeneration, being cut off from any nutrients. And so the cartilage cells die. Maybe there's a little bit of matrix left, but that'll all be cleaned up because osteoclasts are going to come in with the periosteal blood vessel and chew up the matrix, get rid of it. And osteoprogenitor cells are going to come in and start laying down the nice spongy bone that we see, as well as probably some compact bone, but nonetheless, they'll keep the cavity, but they'll make the borders of the bone thicker with spongy bone. So that's what the osteoprogenitor cells that turn into osteoblasts do. Then a secondary ossification center forms at each end or each epiphysis of the bone, and the same process occurs. The only thing that remains is this epiphyseal plate that's hyaline cartilage, and it persists into our late 20s. So the next slide is really an animated video that I cannot show you while taping of endochondral ossification. We first start, I'm going to just go through it. It's just a review again, start with a model of hyaline cartilage. And that model is growing in two directions, okay? And a bony collar has formed. Now the bony collar is going to lead to cutting off nutrients to these cells. Those are chondrocytes, okay? But a primary ossification center is going to form, and we'll have osteoblasts there. Calcification occurs, and we eventually get a death wave of the chondrocytes. But, you know, the wave of death happens after the wave of growth. So if I erase this for a second, and you think about it, the chondrocytes were growing, they're multiplying, they're making this bigger, and that keeps continuing, and it goes this way. But the wave of death, the wave of death sounds terrible, but the wave of death follows that. So the wave of death comes afterwards, and it follows. Then the bone cells make spongy bone. Osteoclasts make sure that the medullary cavity is the right width to accommodate bone marrow as well as blood vessels, and then the secondary ossification centers occur in the epiphyses. And that's the end of bone development. Thank you very much.